Hello everybody, I'm really glad to present you some of my PhD work today concerning the effects of thorium on periphytic biofilm community. This work was realized between two laboratories, first at INRS in Quebec and then at ISTA, now renamed INRAE Bordeaux, under the supervision of the Professor Fortin and the co-supervision of Soisic Morin. Jackie Verdren, Mariam Fadlaoui, and Laura Malvezin participated also in this realization of this work. So to begin, thorium is a tetravalent metallic element belonging to the actinide family and found almost everywhere in the Earth crust, associated with silicates, oxide, and phosphates. That's why, even if thorium is not extracted for itself, it can be released in the environment following uranium and rare earth element mining's activity. Thorium can be also released following the use of phosphate fertilizer for the same reason. So, how can we assess the contamination risk associated with thorium? First, we can really realize physicochemical measurement, which consists to sample the water and determine the contaminant concentration, as well the principal characteristic of the water. And with that, we can use biomonitoring tools, which consist to follow the presence and the health of local species. And we are particularly interested in biofilm as a biomonitor. Periphytic biofilm is a community of microorganisms growing on emerged substrate and inside a matrix of exopolysaccharides secreted by some of them. These microorganisms can be phototrophic and heterotrophic, such as the fungi and bacteria, but also as small grazers like rotifers and psyllets belonging to the micromeiofauna. So why can we use the biofilm as a biomonitor? First, because it can integrate the contamination over the time, whereas the physical-chemical measurement can miss contamination events, and because the biofilm reacts to the total toxicity of the water, even if it contains more than one contaminant. And to finish, biofilm also integrates environmental variations such as the food availability, light, and temperature variation. But what is really interesting with biofilm compared to the other biomonitors is that the biofilm is ubiquitous and you can find it almost everywhere in the earth, even in really extreme environments such as cold Arctic water or really acidic water in Yellowstone, for example. So the objectives were to determine if the periphytic biofilm can accumulate thorium and if the thorium contamination can impact its taxonomic composition and its nutritive characteristics. To do so, we sample natural biofilm and we put it 1 month to colonize in a laboratory channel. Then, 2 days prior the beginning of the exposure, we pre-equilibrated the adsorption sites of our channel to avoid the thorium depletion in the exposure media, and we placed our biofilm site in 3 different conditions. The control condition, 1 nanomolar and 10 nanomolar thorium conditions, during 1, 2, and 4 weeks. After that, we determined the taxonomic composition of diatoms and micromeiofauna by microscopic analysis. And for the bacteria, we used the rRNA 16S gene analysis. We also measured thorium bioaccumulated by the biofilm, and we kept some slides of biofilm to realize a trophic transfer experiment, which I will present you later. So let's begin with the results. Here we have a total number of individuals for the diatom in pink, for the micromeiofauna in blue, And for the bacteria in green, it's expressed in 16s copy number per nanogram DNA. So we see that the three components we observed during this experiment reacted to the thorium exposure at 2 weeks. The number of diatoms was significantly lower than in the control compared to the thorium condition, and we observe the same pattern for the micromeiofauna individuals number. To the contrary, for the bacteria, we observe the opposite. The 16s number were higher in the control condition than in the thorium conditions. Now, concerning the relative abundance of the bacteria genera we observed during this experiment, only 2 of them reacted significantly to the exposure. First, it was Pseudanabena, which is a cyanobacteria also observed during the microscopic analysis, and Sphingopixis, which is a proteobacteria. Pseudanabena was the most abundant genera at the beginning of the experiment, but after 2 weeks of exposure, this genus was not abundant anymore in the control condition, and it was replaced by Sphingopixis. We also observed the same pattern for our 2 thorium exposure, but it was less pronounced as what we have seen in the control condition. So now for the diatoms relative abundance. The dominant species was Agnanthidium exiguum in all conditions, but this diatom was significantly more abundant in the 2 thorium conditions compared to the control. Interestingly, it's known to be found in freshwater of bad quality and to be promoted under combination of copper exposure and high temperatures, which is coherent with our results. Concerning the relative abundance of Micromeiofauna taxa, the flagellates were the most abundant taxon, but did not vary significantly to the thorium exposure. Only 2 taxa reacted to this exposure, Aspidisca cicada, which is a psyllid, and rotifer member of the Phyllodenae family. Here you have Aspidisca cicada and the rotifer individuals number per milligram dry weight of biofilm, and we see that at 2 weeks of exposure, both of them were positively impacted by the thorium exposure. Let us remember the number of the 16s gene copy which I presented you before. Here it's an histogram, but it's the same data. And we can observe an opposite pattern, so the thorium could have an indirect effect on micromeiofauna through direct effects on bacterial community. So to conclude this taxonomic analysis part, we have seen that the thorium can impact the number of total individuals in the different microorganisms communities observed, and that the thorium can also impact the taxonomic composition of these communities by direct effects, like for bacteria, and indirect effects, like we saw for the micromeiofauna. So now let's talk about the bioaccumulation of thorium inside the biofilm. We can see that the bioaccumulation is significantly higher in the 10 nanomolar condition compared to the control and since to the first week of exposure. For the 1 nanomolar condition, it was more mitigated, and the results were significantly different from the control only at 1 and 4 weeks. So now let's talk about the trophic transfer experiment. To realize this experiment, like I said previously, we took biofilm slides from the control and the 10 nanomolar condition after the 4 weeks of exposure, and we gave this biofilm during 4 weeks to the grazer Lymnaea. After these 4 weeks of food exposure, we determined the thorium bioaccumulated in the snail's tissue and also the fatty acids profile of the snail, as well as the fatty acids profile of the biofilm, which we gave as food. So here we have the biofilm in snail's tissue, and we see that the snail fed with thorium-contaminated biofilm presents significantly higher thorium concentration. Concerning the fatty acid profiles, here we have the sum of saturated fatty acids, monounsaturated fatty acids, and polyunsaturated fatty acids as a percentage of total fatty acids in the biofilm. None of the fatty acids categories varied significantly, but if we look in details at the profiles, we can see that some of the fatty acids were significantly lower in the thorium condition than in the control, like this omega-6. For the snails, we can see that the polyunsaturated fatty acids were significantly lower compared to the control, and especially for these omega-6 fatty acids and these omega-3 fatty acids. And this decrease could be linked with lipid peroxidation induced by the thorium. So to conclude this trophic transfer experiment, we have seen that thorium can be bioaccumulated in the biofilm, leading to a decrease of some fatty acids such as this omega-6. And when this biofilm is given as food to the grazer, we have seen that the grazer can also bioaccumulate the thorium. The food exposure also leads to a decrease of polyunsaturated fatty acid in the grazer, leading to changes in fatty acid profile on the both. So now, to conclude, we have seen that thorium can induce changes in community structure, that it can also lead to a loss of its nutritive quality, and that thorium can be bioaccumulated and transferred to the grazer. So here are some questions I ask myself for the future. What about the impact of thorium on biofilm function? Does the bioaccumulated thorium in snails impact their health? And is the bioaccumulated thorium trophically transferable from the snails to their predators? Before I finish, I really want to thank Professor Fortin, Professor Langlois, and Professor Lavoie's research teams, as well as the technical team of INRS, and also Ecovea team from INRAE Bordeaux. And I want also to thank you a lot for your attention.